in and then talk about everyone's favorite topic on drug pricing. So Pushpa, the floor is yours.
Good morning, everyone. Thank you, Dr. Howard. I mean Chip. I get into trouble each time I call him Dr. Howard, but I try my best. I'd like to thank the organizers for the opportunity to present the results of this study. So I will be talking on behalf of my co-primary investigator, Dr. Sanders, Dr. Guptill, the Duke Clinical Research Institute primary investigator, the PROMISE MG study group, and our patients. And I'm going to present the results of the primary analysis of PROMISE MG, which Dr. Howard just, as Dr. Howard just said, is a prospective multi-center observational comparative effectiveness study of MG treatments. Let's see if I get this right. Okay, so these are the 19 study sites. So we had 2 sites in Canada and the rest were in the United States. And on the second column there, you will see the list of the site primary investigators to whom we are very grateful. Without them, this couldn't have happened. And those are my personal disclosures. The specific aims of PROMISE MG were twofold. The first was to compare patient and clinician-reported outcomes in patients with myasthenia gravis who receive azathioprine versus those who receive mycophenolate. The second aim was to compare the same outcomes in patients with myasthenia gravis who receive an adequate dose of these drugs for an adequate duration of time, compared to those who received these drugs at less than adequate doses for less than adequate durations. We defined adequate dose and duration of these 2 drugs based on the definition in the International MG Consensus Guideline published in 2016. And this was what we used. For azathioprine, we defined this, and this was defined actually at the end of the study after the database was locked. And so this was for azathioprine, 2 milligrams per kilogram or more per day for at least a 12-month period. For mycophenolate, at least 2 grams per day for at least 8 months. We also did an exploratory analysis for azathioprine at a dose of 1 to 2 milligram per kilogram per day, because when we went back into the database and looked at the data, there seemed to be a fair number of patients who were treated with that dose. Our inclusion criteria were adults with acquired autoimmune myasthenia gravis, who were drug naive. So this is a new user design in comparative effectiveness. So we chose patients who were not at that time on any immunosuppression and had not previously been treated with any immunosuppression for myasthenia gravis. Those who had received steroids for a non-myasthenia gravis indication over 3 months prior to their baseline visit were allowed to be included. Those who had received steroids for a non-myasthenia gravis indication less than or equal to 30 days, a month before the baseline, were excluded. And those in between were really adjudicated. There weren't a large number of these, but we adjudicated them at the primary investigator level on an individual basis. And if they were on pyridostigmine, as long as the pyridostigmine had been started less than or equal to 90 days prior to the baseline, they were allowed to be enrolled. We had 2 primary outcome measures. The first was a patient-reported outcome measure, and that was a change in baseline on the myasthenia gravis 15-item quality of life score, the revised version. So that's the 3-option version. And we chose a clinically meaningful endpoint as a 5-point change. And if anyone's interested at the end, I can talk about why we chose that number. This has not yet been established formally. The second primary outcome was a clinician-reported outcome. And so that was a composite outcome. And the composite outcome, the two components were to reflect improvement in the disease status, as also reflect the degree of side effects, because we felt that the treatment burden and the degree of adverse events was an extremely important outcome for patients. So, and that included, achieving the minimal manifestation status that sort of looked at the effectiveness part of it, and then no more than grade 1 of the common terminology criteria for adverse events, looking at adverse events. So, and the grade 1 CTCA means that the adverse events are mild, or patients are asymptomatic from them, maybe a mild elevation of transaminases, for instance, from which patients are asymptomatic and no intervention is required. We had these outcome measures as the secondary endpoints, and we chose these because they are used regularly in clinical practice, since this was an observational study in routine clinical practice. We also had, and you'll see here the clinically meaningful changes, and these are from the literature. We also added a secondary outcome of evaluating the number of hospitalizations for myasthenia gravis, because as we developed or sort of did some work with patients, a focus group, a survey, etc., to try and identify the best patient-reported outcome, or to look at these outcomes and see what really reflected patients' desire for an ideal outcome, we heard from them that avoiding hospitalizations was a really big deal. So we added that as a separate outcome measure. In terms of the statistical analysis, we used propensity score weighting to estimate the average treatment effects on the two groups. We estimated the balance between covariates using standardized difference between groups. And in terms of the comparison between groups, we used propensity score weighted linear regression models with several covariates that I'll just talk to you about. So these were the covariates. You can see that we have a couple of, I'm so sorry. We have a couple of covariates that look at the demographics, sex and race. And then we have several covariates that look at common comorbidities that, as you all know, influence our choice of treatments in the clinical setting. We looked at disease parameters, so MGFA class at the start of the treatment. So this was not at the end of the study. We didn't wait to see what happened to them. This was right when they entered and started the treatment. And we dichotomized it into ocular versus all generalized. And then, of course, the antibodies, thymoma, and the late onset versus early onset myasthenia gravis. We also looked at the treatment with prednisone. We used the area under the curve for obvious reasons, and BMI as well. We were planning to include these covariates as well, but we didn't have enough numbers, or we had no patients who were pregnant or lactating, for instance, so those were not included. So this is it's a little bit hard to see, and I'm sorry about that, but I'm going to try and take you through the study flow. So I told you the inclusion criteria. So we were planning to enroll 220 patients, to have 200 with a 10% dropout rate for the analytic data set. And we ended up including enrolling 167, so about 76% of our planned enrollment. And some of the reasons for not being able to enroll our numbers were the fact that we use the new user design. And in many tertiary care centers, patients are referred from the outside, and they're often on pyridostigmine and very often also on prednisone. So anybody who was on prednisone was automatically excluded. The other point that we found was, at the end, we found that we had enrolled almost 40% of these patients, of the 167, were ocular myasthenia gravis. And as you know, ocular myasthenia gravis may not require azathioprine or mycophenolate. And sure enough, only 25%, about 17 of the 67 patients, actually received azathioprine or mycophenolate and so could go into the primary analysis data set. The rest could not be included. So, and what did we do with the 167 who were included? We had to cull out those who were treated with azathioprine or mycophenolate, and 34 were treated with azathioprine and 48 with mycophenolate. So that 82 formed the primary analysis data set. The other 85 were excluded, and we have a whole bunch of exploratory analysis that are being performed. So let's look at the demographics. This is the full group of 167. We had almost 2/3 male, and a large proportion were white. And in 89%, the disease onset was after the age of 50, again, sort of similar to the Italian registry cohort that we heard. You'll see here that the mean age at the initial visit of the whole group was 63, and it didn't really differ very much when we stratified by sex. And the mean age at the initial visit was 65, again, not very different when we stratified by sex. The mean duration of the illness was 1.2 years, and the median was 5.2 months. The IQR is there. In terms of the MGFA class at entry, I spoke about the 40% being ocular. About 38% were class 2, and another 20% were class 3. So class 2 and 3 formed 58% of our patients. We had a couple of patients in class 5, and those happened because in terms of the IVIG and the plasma exchange, we said that if they were admitted to the hospital, drug naive, got IVIG or plasma exchange, as long as we could gather the outcome measures within 24 hours, we were okay to include them. So we had a couple, not a whole lot though. So let's look at the azathioprine versus mycophenolate cohort. So as I said, we had 34 patients of the 82, so 41% on azathioprine, and a slightly larger proportion, 59% on mycophenolate. There were 11 patients who received both, so they crossed over from one treatment to the other. And in these patients, we used an intent to treat paradigm and enrolled them in the group of the first treatment. So 8 of them received azathioprine first, so 8 of them, so 8 went into the azathioprine group and 2 went into the mycophenolate group. So this is a little bit dizzy, but I'll walk you through it. So this is the baseline characteristics. And because it's an observational study, this is an important slide. This is sort of like table 1, but we want to know the balance between covariates and what happened after propensity score weighting. So you will see here that this is for the categorical covariates. Diabetes mellitus and a history of cancer was higher, I believe, in the mycophenolate group compared to the azathioprine group, and the standardized mean difference was around 0.3 or a little higher than 0.3. As Dr. Banat-Tapia mentioned earlier, you want the standardized mean difference to be less than 0.1. Some people use less than 0.2. I think Dr. Banat-Tapia mentioned 0.2 in order to say that the groups are balanced. And so, but you will see that after propensity score weighting, the standardized difference was 0, and all of these covariates were then perfectly balanced. So it's sort of, you're sort of forcing it into a data set that resembles that of a randomized controlled trial where the baseline covariates are equal. So looking at the continuous covariates and the baseline outcome measures, you'll see here, many of them were close to 0.1, maybe slightly higher, even to start with. And interestingly, all of the baseline covariates were really reasonably well balanced, even at the outset. And with propensity score weighting, they were all well balanced, perfectly balanced. So looking at the overall population, so this is an unadjusted analysis. How many people in the azathioprine group versus the mycophenolate group actually achieved meaningful changes in the outcome measures? So you will see that a large proportion of patients in both groups achieved meaningful changes in all of the outcome measures. But you'll also see that the difference was statistically significant, favoring mycophenolate a little bit. So a slightly higher proportion of patients on mycophenolate achieved meaningful changes in these decreases in the outcome measures compared to azathioprine, for the physician-reported outcomes, but there was really no difference statistically in the patient-reported outcomes. So then let's look at the linear regression, adjusted for propensity score weighted and adjusted for all of the covariates that I mentioned. So there was a approximately 3-point change in the greatest change from baseline between the two groups favoring mycophenolate on the primary outcome of MGQOL-15. And that change was statistically significant. But as you may remember, this change did not meet our definition of a clinically meaningful change. However, when we looked at the number of participants, so the proportion of participants in both these drug groups with a meaningful change, there was approximately a 7% higher proportion in the azathioprine group who actually reached that point, but that was not statistically significant. For the co-primary outcome measure, the composite outcome measure, approximately 2% of patients in the azathioprine group achieved, greater patients in the azathioprine group achieved this measure, but that was also not statistically significant. How about all of the secondary outcomes? Again, for the greatest change from baseline, a 1-point change in the MG-ADL favoring mycophenolate, a 15% greater proportion of patients with a meaningful change on MMT favoring azathioprine. Those were the two that were statistically significant. This 1-point change, again, did not meet our definition of clinically meaningful. How about other outcomes? We talked about hospitalizations. There was really no difference between hospitalizations in the mycophenolate group and the azathioprine group. This is hospitalizations for MG alone. And this is interesting. We found this is a mean time to achieve the lowest MGQOL score between drugs. Now, it is not the time from the point when the drugs had been maximized, so not at the optimum dose. It's the entire time. And you will see that, surprisingly, mycophenolate took 17 months with a range of 1.2 to 38 months to get to that point. Azathioprine, 15 months, range of 1.6 to 33 months, and the difference was not significant. And the difference is, I think, not significant because of this result with the mycophenolate. And we will look a little bit more in detail at this number. I don't have that data quite yet, and I don't also have the median number of months. But this, I think, is in contrast with our suppositions when we did the randomized controlled trials of mycophenolate in terms of how quickly it acts. But I do think it's in keeping with clinical practice, and I'd like to hear your thoughts. How about safety of these two drugs? Approximately 32% of patients on mycophenolate and 19% of patients on 32% of patients on azathioprine and 19% of patients on mycophenolate had side effects. The side effect profile was really in keeping with what we know about these drugs, transaminitis, and hematologic side effects, and the influenza-like delayed hypersensitivity reaction in the azathioprine group, one pancreatitis as well, and mostly GI side effects in the mycophenolate group. The risk difference was 14% and favoring safety of mycophenolate over azathioprine. But the confidence interval, as you can see, is quite wide, reflecting the imprecision of this analysis, likely the small numbers. But the number needed to harm, I mean, at the end of the day, significant or not, I think safety is one parameter for which we have a lower threshold. The number needed to harm was 8, favoring mycophenolate. How about the second aim of adequate dose versus duration and duration versus not? So again, this is the baseline characteristics. There were more men and more generalized myasthenia gravis patients in the group that did not receive adequate dose and duration. But all of these were perfectly balanced after propensity score weighting. Similarly, for the continuous covariates and the baseline outcome measures, again, you'll see a fair balance of the outcome measures at baseline, quite surprisingly, but they're all balanced. There was not much difference to start with, and they're balanced after weighting. Again, looking at this overall population, unadjusted, you will see that both the group of patients who received adequate dose duration versus not had a large proportion had meaningful changes in all outcome measures. None of these differences between the two were statistically significant. And in terms of looking at the a regression model with the covariates and propensity score weighting, there was no difference in the primary or the secondary outcomes between patients who received adequate dose and duration versus not. When we looked at the 1 to 2 milligram, when we added the patients who had received between 1 and 2 milligrams per kilogram of azathioprine to the 2 milligram per kilogram and above group, we had about 7 extra patients. And again, you see the same pattern. Not a lot of difference in terms of achieving meaningful outcome, meaningful changes in outcome measures, reductions, and no differences in the primary or secondary outcomes. So what can we take home from this? In terms of effectiveness, most of the 82 patients who were treated with either azathioprine or mycophenolate had clinically meaningful changes in all outcome measures, regardless of the drug, and whether or not they received what we considered an adequate dose and duration of the drug. There was a small statistically significant difference in the primary outcome measure of change, greatest change from baseline, drop from baseline in the MGQOL-15R. But this did not meet our definition of clinically meaningful, and we can talk about that. The time to response for mycophenolate was longer than we usually teach or we usually read. Adverse events were more frequent and more serious in patients receiving azathioprine. And is it possible that a lower than recommended 2 milligrams per kilogram dose of azathioprine may be useful in treating patients and perhaps even mitigate some of the side effects in clinical practice? And so I think, and we're looking at that data a little bit more in depth, and I think really the choice of treatments would be influenced more by the side effect profile than by the effectiveness, at least with this data set. The limitations of the study are many. It's an observational study. And we wondered obviously about generalizability. And of course, when it comes to the age population, the same question that we had in the discussion earlier today comes up. So the study was carried out in tertiary centers. I'm not quite sure how to explain that age difference because of a tertiary center, though, obviously. I don't quite understand it because I can't imagine that I would only have older patients who were referred unless they were all so sick because of comorbidities that, you know, my colleagues in the community didn't want to take care of them. I find that hard to sort of believe. We had a lower sample size than anticipated, and that probably affected our ability to detect differences because of the imprecision of the data set. And you will see that in the confidence intervals that are fairly wide and sort of cross, this clinically meaningful and unmeaningful areas. Propensity score weighting does not really correct for unknown confounders, so we don't know what we've missed. The number of ocular MG patients was higher than anticipated, although it's an interesting data set on whom we'll be able to do some exploratory analysis. The COVID-19 pandemic affected follow-up, especially in terms of getting the clinician-reported outcome measures on telephone visits, and so we had missing data on those, contributing to the imprecision. And that was my last slide. And this study was funded by the Patient-Centered Outcomes Research Institute. And we're very grateful to the Myasthenia Gravis Foundation of America for the support of preliminary work in sort of identifying a relevant patient-reported outcome measure for the study. And that paper is published in Muscle and Nerve. I think we'll take questions last. Those are my grandcats. So.
Thank you. Thank you, Pushpa. Our next speaker is Gordon Smith, and he's going to talk about drug pricing.
Right, welcome everyone. Let's see this. I do have slides. There we go. Fantastic. Well, thanks everyone. It's great to see such a good turnout here, given the lovely weather outside, and I really appreciate the invitation to talk about drug pricing. Chip, I didn't realize this was everyone's favorite topic, but it's an important one. And what I really hope to talk to you about over the next 20 minutes, why is this important? Why should you care about it? What's driving it? Why it's particularly important for all of us in the room and the patients that we care for, and then what can we do about it, both in terms of a health system and society, and what can we do about it for individuals. These are my conflicts. I do consulting with both argenx and Alexion, and as you might figure, they are part of this story, which really has to begin looking at healthcare value at a large level. And so this figure shows you the, whoa, I'm having a hard time controlling these. Let's go back. Here we go, the wrong green button. So this is healthcare spending per capita relative to life expectancy. And as you probably all know, the United States has by far and away the most expensive healthcare system in the world. And we are not getting what we pay for based on this curve. You'll see we're very expensive, but our quality is well below most of the rest of the industrialized world. And one can see similar sorts of data about infant mortality and others. And so we spend a lot and we don't get quite what we need out of it. And this cost continues to go up, and it's really being driven by two things: hospital-based care and prescription drugs. Now, these aren't the percentage of costs attributable to those. These are the percentage of change. Those of you who practice in the United States, I think will find it unsurprising that we've actually become a little cheaper as providers, but hospital care and drug prices continue to go through the roof. And this is a recession-proof business, as it were. So this shows you the average year-over-year inflation rate for brand name drugs, top 260 products, relative to the inflation rate for any given year. And so you'll see, even in years, and I'm sorry, I can't really point over here, I'll try. You see here in the Great Deflation or Great Recession, there was a little bit of deflation, yet drug costs went up 8%. And even in the last couple of years where the rate of cost increase has gone down, it's still well outpacing inflation. It'll be interesting to see what it looks like given where inflation is right now. But this has been a trend for some time. And this in turn leads to something that's been referred to in the cancer literature as financial toxicity. And we've heard a lot of talks here at this meeting, and including Pushpa's talking about adverse effects on our patients from the drugs we prescribe. I will posit that most of you that practice here in the United States see financial toxicity every day. And if you don't, you either are in perhaps a concierge practice or you don't ask your patients about this, but we certainly see it every day where I practice. And just some metrics about this: 1 in 4 Americans have difficulty affording their medicines. 15% of poor Americans ration their medications. About a third of Medicare beneficiaries take fewer doses than recommended, and about 20% of prescriptions are abandoned at the pharmacy counter. And there's some really interesting data on this from the oncology literature. So these are prescription abandonment rates for patients who are taking novel oral anti-cancer drugs. So these drugs are really meant to modify the disease course of malignancy, often to prolong life or certainly improve their quality of life. And you can see even for inexpensive drugs, $10, about 10% of prescriptions are abandoned at the counter. So maybe that's not a financial toxicity, maybe that's some other variable. But when we look at expensive drugs, and this is over $2,000, you'll see half are abandoned at the pharmacy counter. And these are the drugs that are most impactful, or thought to be most impactful. And for anyone who's had cancer or cared for cancer patients, the idea that half of them are not filling this kind of medication is really striking evidence of financial toxicity. And if you ask your patients about this, you'll hear it all the time. So why are drugs expensive? This is a really complicated question that I'm not competent to answer, but I can provide maybe a satellite view. And one reason is that it's not a typical free market economy. This does not look like the process that your washing machine or your laptop computer traverses from manufacturer to your household. And there are multiple reasons for that. One reason that receives a lot of attention is the pharmacy benefit manager, which sits in the middle, and PBMs negotiate discounts on either end of the deal. I'm not here to vilify PBMs in any way, shape, or form, but this economy doesn't make sense as a rolled up entity. And it's not surprising that at each step, the players behave, I think, pretty much like you would expect them to behave. Now, there are, of course, other variables involved, but the bottom line is that drug costs are primarily driven by the pharmaceutical companies and drug manufacturers. And so you can see PBMs are over here, and actually provider-based expenses are greater than PBMs, but it's really drug manufacturing, which is where we really should focus our attention for the rest of the time. And there are several areas in the market that really are driving this. One that I really want to focus on today are specialty and orphan drugs. And obviously, myasthenia gravis, Lambert-Eaton syndrome, congenital myasthenic syndromes, these are all orphan diseases, which I'll define in a moment. But these are really driving the escalating drug costs in the United States. And for those of you who care for or are familiar with the story of MS, you see a really striking example. These are MS disease-modifying therapies and their costs over 2004 to 2016, not 18. And just one great example is Copaxone, released in 1996 at about $8,000. Same drug now costs over $80,000, 1,000% list price increase. And so this is a train wreck. I think we all agree about it. It's received a lot of attention, yet no relief so far. I think this is a story that I sincerely hope does not replicate in the really exciting therapeutic era we have in myasthenia. Just by comparison, these are relatively inexpensive drugs. The only one that went up actually is pregabalin, and this addresses another concept that is the tactic of sort of play for delay or other strategies that delay market entry for generic manufacturers. And I'm not going to get into that. It's actually a really interesting topic. And so what is an orphan disease? Well, it's a disease that affects, I'll show you in the next slide, by definition in the United States, less than 200,000 people. There's some other attributes, but rare diseases are actually common. About 10% of the world has a rare disease. In the United States, that means over 30 million people. That means rare diseases are cumulatively more common than diabetes, heart disease, or cancer. Diabetes is catching up, but these are very common. And for those of you who are child neurologists, you'll recognize that over a third of deaths in the first year of life are from rare diseases. So these diseases disproportionately affect children, who I think are often, and we learned a bit about this today, disadvantaged in our health system and discovery. These drugs are expensive. And so this shows you the average cost of an orphan product, top 100 products versus a non-orphan product. So about $150,000 a year versus $33,000 a year. I actually remember when we thought that was expensive, but it pales in comparison. And you can see here the trend over time in orphan drug designations, which are the blue lines, and the red lines or orange are actually approvals. And so orphan drugs comprise a disproportionate percentage, and in some years, even a majority of new drug approvals. This shows those figures. So the dark blue, year by year, are non-orphan product approvals, and the light blue are orphan product approvals. So you'll see in 2018, there were 9 more orphan drugs approved by the FDA than non-orphan drugs. And this cost adds up over time. So this actually shows worldwide data in terms of the increase in cost of orphan products. So you can see it's projected to continue at about 11% compound annual growth rate. You can see the total cost in the billions, and the percentage on the right of total pharmaceutical spend projected to be about 17%. Again, disproportionate relative to the frequency of these disorders. So what is an orphan drug defined as in the United States? And so it was defined by the Orphan Drug Act, which was passed in 1983, Senators Waxman and Hatch. And before this, there had only been 10 orphan products or rare disease products approved in the United States. And since that time, it's obviously turned around. I've shown you evidence of that, over 600 approvals. So it's certainly been an incentive, and I'll show you why in a second. But the definition, as I've alluded to already, 200,000 people or less with the disease, a scientific rationale. There is another pathway to orphan designation, which is uncommonly used, but sometimes which is evidence that sales won't cover the development costs for the drug. But usually the designation is based on the 200,000 people with the disease, which is a pretty liberal definition, I would say. And there's some fairly common diseases that impact 200,000 patients in the United States every year. And so these are the benefits from the Orphan Drug Act. There are tax benefits. There is a waiver of the FDA approval fee. The review time is abbreviated. But the meat of this, and really the true incentive, is in the exclusivity period. So a typical non-orphan product has an exclusivity period of 3 to 5 years, orphaned products 7 years. There's a whole other narrative around patents, which I won't get into, which is super complicated. So the more you dig at this, the more it's really interesting. And there's these crevices that problems can crawl through. But it's really the exclusivity period. So this is a system that is ripe for gaming. And I'm going to show you evidence, give you an example that you already know about that is actually a gift that keeps on giving in terms of understanding this problem. And then I'll show you about the system level. But I do want to talk about Ted Burns, who we all know, who obviously can't be here today. He's well aware of this talk and has been texting me, which is great. But Ted has identified a number of ways that the Orphan Drug Act can be perverted, frankly. And I'm just going to cover 4 of them here and give you an example. So 1, purchase the rights to an existing inexpensive unapproved drug, and then you can aggressively monetize it, repurpose an existing inexpensive approved drug for a different indication. So-called Trojan applications, which mean you hide the common disease within the rare disease horse, so to speak. So get orphan drug approval for a rare disease and to go big after you price it high. Or salami slicing, take a common disease, slice it up into little teeny bits of rare subtypes, and then you can go big with the common disease. These strategies are all particularly attractive in the rare disease market because the therapeutic space is usually not crowded. You have a limited marketplace, and we see this actually in lots of areas in neurology. So the story, not surprisingly to anyone in this room, that is a great example of this is 3,4-diaminopyridine. For the 2 or 3 of you who don't know this story, for several decades, the base formulation of 3,4-DAP pictured here was available in the United States essentially for free from Jacobus Pharmaceuticals or at low cost from compounding pharmacies. The phosphate salt has been developed as FIRDAPSE licensed in the United States by Catalyst Pharmaceuticals. And I think we all know the story, so I'll go through briefly. About 7 years ago or 6 years ago, there was a great deal of concern about this. And many of you in this room, I can see you by face, participated in this editorial that Ted and I pulled together for Muscle and Nerve. I'm going to come back to this in a moment, but this editorial has had a huge impact on the narrative, the national narrative, the public narrative, and the political discourse in this space. There was a lot of coverage about what we feared would come and ultimately did come. This is one of my patients, Fred Kellerman. I can talk about him because he was on the cover of the New York Times talking about this. There was coverage in NPR, the NewsHour and whatnot, and I will spare you the long story. We can talk more about it if you like in the question and answer, but the unthinkable happened, that the phosphate salt was approved at an annual price of $375,000, which was beyond anyone's wildest fear. This story is actually very fascinating, but I'll get to the end because there is an update on this story that dropped into my email just about an hour and a half ago. One that you may have heard is patients are actually expressing concern. This is a great article by Ed Silverman about not being able to access the base formulation, which they felt worked better. There's been discussion about trying to figure out how quantitatively large this group is, but the update is there's now legislation that's been proposed by Senators Cassidy and Baldwin, so bipartisan legislation that would actually fix the legal issue that led to the Jacobus product being pulled from the market. So very interesting. We can talk more about that in the question and answer if you like. Now, that's a scary story. Is it unique? Well, it's not unique. There's certainly other bad actors out there. You read about them in the newspapers and see them on TV. But as a class or a mechanism of approval for orphan drugs, this, I think, tells the story. And it's all the way from the beginning of the Orphan Drug Act. The red approvals here are new molecular entity first in class. These are highly innovative, or most of them are highly innovative. This is the therapeutic development that was intended by Senators Waxman and Hatch, and Senator Waxman has actually attempted to update the ODA without success. The sort of salmon color here are previously existing drugs approved for a new orphan purpose. And you can see the large majority of drugs being approved under the ODA are for this reason. This is not what the Orphan Drug Act was intended to do. This gives you some numerical data and a little more data more recently, going up to 2017. But you can see the top two columns here are secondary indications and a new formulation, which is actually a part of the way companies delay generic entries into the marketplace. And so this adds up over time, right? Rare diseases are common. And when I first published this, people thought I was crazy by suggesting, what if 10% of them were treatable? I don't think anyone thinks that's crazy now. It's really exciting. And we're all excited by the therapeutic development in myasthenia and in genetic neuromuscular diseases. But in a world where 10% of these diseases, or rather 30% of these diseases, are treatable, the percentage of the gross domestic product of the United States that would go towards funding drugs for rare diseases would be 7%, which is astronomical. It's so large, it's kind of hard to put your mind around. So I've tried a few. This is my most recent one. You can tell me if it works. If you look at gross domestic product by state, one would be $1.5 trillion. Now, these are in 2019 dollars. That would basically be the entire economic output to which we're contributing in Florida, which I think is the fourth one here, plus Virginia, where I live. And so the good citizens of Florida and Virginia would be contributing their entire economic productivity towards funding rare disease drugs, which is not a sustainable process. And of course, this raises another ethical question about distributive justice, or another way of looking at it, spinning it around, is opportunity cost, right? So we can take an expensive drug, treat one patient, or we can use, in this case, $750,000, which of course comes from the first year of nusinersen, and treat 1,000 people with $750 of care. And ultimately, it's a zero-sum game. We can't print more money just to fund this. And in the United States, people are dreadfully afraid of the term rationing, yet rationing is happening in our healthcare system right now. Financial toxicity is the symptom of rationing. Our rationing is not explicit rationing, it's implicit rationing. And the people who are suffering the consequences from this crisis and opportunity cost of distributive justice are the same people and populations that are disadvantaged and suffer from healthcare disparities. And if you don't see it, come visit me in Richmond and come to clinic, and I'm happy to share some stories with you. So this is important. So how do we get out of it? And we are going to hear a great talk about ICER and value in a moment. And I would propose, and I'm going to briefly touch on this, that I think value is what's going to set us free, which is really quality and service over cost. There's a lot of discussion about fairness in drug pricing, which is a little different. And there are variables in fairness beyond quality, which are development costs. What about the externality? Is the first FcRn inhibitor approved for human use more valuable? Should it cost more because it's the first one ever approved? The innovation, is there value in that innovation? And obviously shareholder return. And so these are interesting topics to debate, but I think value is really where it's at. And most countries in the world have a governmental health technology assessment organization that determines the value or estimates the value of a new treatment And here's where the really important point. They don't say yes or no. They say, this is what we think the value is, and then they negotiate. And I think a lot of the controversy around ICER misses that last point. So this is NICE, and I'm not going to say how great NICE is, how nice NICE is. Our colleagues from the UK can tell us the problems with NICE, and there are problems, but they do important work. And in the United States, our NICE equivalent is ICER, which it's interesting to see ICER is it president, CEO? The grand poobah at ICER, Steve Pearson, who's really brilliant. In the cover of the Washington Examiner in 2018, this obscure research firm, I don't think anyone now, 4 years later, is referring to ICER as an obscure research firm. It is the de facto HTA for the United States. I have to, every talk I give now, I have to plug the neurology podcast for TED. And so if you're, it's great, you should check it out. But if you're interested in drug pricing, there are several podcasts that Steve Pearson has done that are really informative and instructive for our topics. And I won't go into the process, we'll hear about that in the next talk, but I'm sure most of you are familiar with the recent ICER analysis of the new treatments available to us, or two of them, for myasthenia gravis. And they basically use a structured approach for drug valuation. The quality adjusted life year is the point of the realm, which is imperfect. And they then calculate using a rather complicated process that does include contextual variables, what the discount would need to be to achieve a value-based price. It's worth pointing out that their placeholder price for our efgartigimod was higher than the ultimate average sales price, which is refreshing. Obviously, the average sales price is still a lot more than the value-based price that ICER estimated. But again, this is the beginning. This isn't the end. And so I think people see this, in particular our colleagues in industry, their blood pressure goes up. But this is really the beginning. And I'll give you a couple of examples of this. One from the UK. So the world's most expensive drug is recommended for approval in the UK. And then also just recently, the other day, ICER released a favorable assessment of Bluebird Bio's gene therapy, which is for beta thalassemia. So why do you tell you about beta thalassemia drug? Well, it's a $2 million gene therapy. Pretty expensive, right? That's right there with Zolgensma. And the reason that this was viewed as a favorable assessment is it was based on an assumption of value-based contracting. And I think this is where the future is going. And there are all kinds of really interesting innovation models happening in this space. This is one, it's from the cancer literature and Pfizer, but it's a warranty. And obviously, this is just putting terminology around a value-based contract that simple people like me can understand. If it doesn't work, you get your money back. I understand that. There are subscription-based approaches, and our colleagues in industry right now are very well aware of this, and they're busy negotiating these value-based contracts. And I will talk a little more about this in a moment after I pivot and talk about what are the system solutions. Well, there are a whole bunch of them that have been proposed, including penalties for companies that raise drugs beyond the rate of inflation, which we've seen has been happening every year for as far back as we look. Capping out-of-pocket costs, very important for our patients, would directly impact financial toxicity. One that is attractive because of its simplicity, and I'll come back to, is international referencing. Why does the rest of the world pay less money than we do in the United States? There's some problems with this. And then intervening in the marketplace to increase competition. And this really is largely focused on smoothing the way for generic entry into the marketplace. So prohibiting evergreening or these sort of pay for delay strategies. There's another tactic that's received interest in the last year or so and periodically comes up, and that's NIH a stakeholder. I don't know about new drug approvals in the last 6 years, but for the 6 years before that, every single new drug approval by the FDA had its original, if not inception, preclinical, early clinical work funded in part by the NIH. And in fact, there was a bipartisan bill called the We Paid Act that proposed to address this. And this has come up again a couple of times in the last year, but most notably in relation to the treatments for COVID-19. And so, and it's coming up again with specific treatments, particularly in prostate cancer. So this is something to keep an eye on. But I'm going to come back to value. This also just is something that Jason Crowell texted me yesterday. So if you're interested in the argument, this is a very brief blog post on the Commonwealth Fund that makes the point that some of these other workarounds, in particular, international referencing, have problems. And I'm probably running over, so I won't get into that. But really, thoughtful approach to value-based pricing is, I think, ironically, where the money's at right now. So it's a bad metaphor, I suppose. So what can we do, right? I'm not a health policy expert. I know my colleague from ICER is cringing with every word I say, but there are things that we can do. So we can talk to our patients about drug costs, not only individually, but there are many patients that are interested in distributive justice. And there are appropriate ways to have these conversations. You know, try to find the least expensive means for them to get their drug. And so GoodRx is a good strategy. Speak up when you can. Editorials matter. They matter a lot. And the article today that was released about the new proposed legislation to address the catastrophe that happened just recently with 3,4-DAP referenced that editorial. That had lasting impact, that editorial. So write, advocate, speak your mind, consider your collaborations. What trials do you want to do? You know, I've run big clinical research groups in the past, and we look for trials that have margin so that we can, you know, further support the research in which we have passionate interest. Be very thoughtful about what's on the other side of that. What's the policy impact? What's the impact on your patients? Raise questions about sponsorships. If there's a bad actor, you know, speak up to your national organization or specialty organization about whether that's a sponsorship that's appropriate. And then I'm not going to have time to get into it, but don't buy into this expanded access. It won't cost your patient anything nonsense. We hear this all the time. What are you worried about? Your patient isn't going to have to pay anything more. Well, where do you think the money comes from? It comes from somewhere, and it comes from all of us, whether it's your insurance premiums, your taxes. This is a real problem. And there's a shady side, a very shady side to some of these kind of patient assistance programs that I won't get into again for limited time. But if you watch kind of health policy or even just the front page of your newspaper, you will see a steady stream of significant fines to industry, just two of which that are within the last year or so pictured here for inappropriate sort of anti-kickback violations in this space. And so don't buy this whole no one pays. Trust me, people pay. And so I'm going to end there. Hopefully that was informative. This is Ted Burns, who's really been an inspiration and thought leader of this. So I want to acknowledge most of these ideas came from in between Ted's ears, sitting in a pose that's familiar to any of you who know him from working at UVA. And in many ways, I am Ted's mouthpiece. So thank you. Thanks. I'm sorry, Chip, I'm sure I've gone over, but there's no timer. So.
Thanks, Gordon. All right, let's move on with our last speaker of the day. We've asked Felicia Abdullah from ICER to come and talk about the methodology, how they approach the decisions that come forth from their institute. Thank you.
Thank you for the introduction, Dr. Howard. And again, thank you to the organizers of this conference for inviting me to be part of this. So I'm really excited to share about our work on assessment of the effectiveness and value for the new treatments for myasthenia gravis, eculizumab and efgartigimod. All right. So to provide just a quick roadmap for what I'm going to be discussing today, for those who don't know about ICER, well, thanks to Dr. Smith, he introduced us well, but I'm just going to say a little bit about our organization, but also provide just a quick overview of how we went about doing our myasthenia gravis review and some of the things we found. And also beyond the final report, we do have a final report on our review, some of the policy recommendations that came out of our public meeting, and even what we do beyond that. So ICER is an independent health technology assessment organization. We develop publicly available value assessment reports that examine the comparative effectiveness and cost effectiveness of various healthcare interventions. Our HTA activities are funded exclusively by the funding we get from nonprofit foundations, and this allows us to be independent in our work. And our goal as an organization really is, we believe that we can use evidence in a transparent way to align prices with the benefits they provide for patients and families. We believe this is the way in which we can help to improve access and affordability while retaining the incentives that are necessary for future innovation. So to be transparent about our funding, again, I said our HTA activities funded fully by nonprofit foundations. We do get about 19% of contributions from manufacturers and health plans, and this part of our funding is fully walled off from our HTA activities, and we use that specifically for an annual ICER policy summit that we put together. So turning to our assessment of the effectiveness and value of eculizumab and efgartigimod for myasthenia gravis, the final report, as you can see for this evaluation, was completed and posted in October 2021. And that was before efgartigimod was actually FDA approved. And this really is in alignment with what we hope to do to be able to actually get the report out there right around the time of FDA approval, so that we can provide the information necessary for decision makers that are going to be making decisions around drug pricing, but also coverage policies for patients. So how do we go about developing our report? As a first stage, importantly for us, we scope patient groups and patients to get a sense of their lived experience with the condition, because we think this helps us to properly frame the question. We also get guidance from clinical experts. We're not experts in myasthenia gravis, and again, we evaluate different conditions. So for each report we develop, we get guidance from clinical experts. We also scope manufacturers and other stakeholders such as payers to get their perspective. Once we do that, we go ahead and do our own evidence analysis. And we partner with academic colleagues specifically for this report. We partner with our academic colleague at University of Illinois at Chicago for the cost effectiveness pricing. And once we have our report, we go to a draft report where we put it out there for public comments, and based on the comments, revise the report. We also do have a set of expert reviewers that also provide comments. And as you can see, two of the presenters that spoke before me actually served as expert reviewers for this report. And once we have the final report, we go ahead to do a public meeting where we get the opportunity to present on the evidence, but also have a policy conversation. And really the goal for us doing this is generally right now, drug pricing, coverage decisions, they're being done behind closed doors where patients and clinicians really do have no say in this. And our goal is to be able to move this conversation to a public setting based on the evidence and the work that we've done. So just to get a sense of how we go about doing our work, we do have a value assessment framework, which really is grounded in our long-term perspective on value. We think about value as a comprehensive, complex process, the foundation really, which is the comparative clinical effectiveness, where we're asking questions around how does this new drug, when compared with a comparator, what is the net health benefit? How does it help the patient to return to function? What are the side effect consideration? And how does it perhaps help patients to live longer life? And once we had the cost consideration to that, which really not just the drug cost, but the total cost, we're asking questions around cost effectiveness. Now, beyond these two core measures, that is really part of our value assessment framework, we also described extensively in the report and as part of our public meeting, what we call benefits beyond health. And this really is in recognition that sometimes there are things that are important to patients that go beyond measured clinical outcomes that we may not be able to quantitatively capture. We also generally highlight special social or ethical priorities that may be important for a disease area. For example, there may be severe diseases that we want to highlight. There may also be conditions with which, if we have a new treatment in that space, would help us to reduce health disparity in the society. Right. So just to provide a brief overview of some of the things we heard from patients at the beginning of this review, a lot of patients told us about the long path to diagnosis and just the impact of living with myasthenia gravis, which can feel like a full-time job itself. And some of the challenges with the current treatment, including the side effects, but also barriers to access that they face as sometimes, especially when they're given particularly off-label treatments. We also heard a lot about the race, ethnicity, gender disparities, and the financial burden associated with this condition and the treatments. So the scope for our review, after speaking with all the various stakeholders, we defined to focus on adults with generalized myasthenia gravis who have failed or are intolerant to standard treatment. And we were looking at the intervention eculizumab and efgartigimod. Of course, in the clinical trials of these drugs, they're used in addition to conventional therapy. And so comparing it to a situation in which patients would have to continue conventional therapy alone, which is standard of care. But based on a lot of things, we also heard that rituximab and maintenance IVIG seems to also be used widely in clinical practice. For these patients, we felt it would be important for us to compare this new interventions with rituximab and maintenance IVIG. But I will say it right here that we found really limited data to be able to do that. And so I'm not going to be presenting results on those comparators. We do have a long list of outcomes that we have in our scope. For the purpose of this presentation, I'm going to be focusing on the measures of treatment response and the QMG and MG-ADL. So we conducted a systematic literature review, and I'm sure many of you here are familiar and know more about this clinical trials than I do. But just to quickly go over this, the main phase 3 trial we identified for eculizumab was the REGAIN trial, conducted in patients with AChR positive antibodies and who are refractory to treatment. Although eculizumab in this trial did not meet its primary endpoint, which was quite conservative, but it showed consistent clinically important improvements in MG-ADL, QMG, and quality of life measure at 4 weeks. Improvements were sustained at 26 weeks and maintained through 130 weeks in the open label extension studies. Serious adverse events were uncommon and comparable versus placebo, although I would note that there appeared to be more discontinuation for patients who were on eculizumab in the trial. Again, just to highlight that there is a black box warning for using eculizumab for meningococcal infection. For efgartigimod, we also identified one phase 3 trial, the ADAPT trial, conducted in a wider myasthenia gravis population of patients with or without AChR antibodies. Also showed significant improvement in MG-ADL, QMG, and the quality of life measure at 4 weeks after the first cycle. You would note that the improvement for this appeared to decline by 8 weeks, and this was really due to the unusual dosing schedule in the clinical trial, where patients were only redosed when they no longer had a clinically meaningful improvement in MG-ADL. Again, serious adverse events were uncommon and comparable versus placebo in the short-term clinical trial. So some of the areas of uncertainty in the clinical evidence that we highlighted in the report was really, it's really unclear if or when to stop this treatment once you start. There were really insufficient data on important subgroups such as seronegative patients, masked antibody positive patients. But we also noticed that for the clinical trials and for eculizumab, for example, they had no Black American patients in the trial. And so we had limited data on non-white population. We had insufficient data on some patient important outcomes that we would have loved to know more about. And again, I spoke a little bit about the dosing regimen for efgartigimod. It's uncertain what exactly is going to happen in clinical practice. If this is going to look like what happened in the clinical trial, or physicians will tailor this differently. So we do have something called evidence rating, and this is really to give a judgment on our take on the evidence based on our evidence matrix. And so here's the evidence matrix. If you're looking from left to right, it's really moving from negative net benefit to substantial net benefit. We quantify this with a letter grade. And from top to bottom there is showing how our certainty goes from being high to low. And so for eculizumab versus placebo, we gave an evidence rating of B+, which really showing that we have moderate certainty of a small or substantial net health benefit. And when we think about the balance of information on benefits and adverse events and the long-term benefits and the adverse events on eculizumab, we know. For efgartigimod, we give a C++, which really indicates a moderate certainty of a comparable, small or substantial net health benefit. Now, I would just note that the more uncertainty that we put in that was really a reflection of the fact that efgartigimod is relatively new. Right now, all we have is the short-term benefits. It's unclear what the dosing regimen will look in the real world and how that may have impact on the long-term safety. So turning to the cost effectiveness, and for the purpose of those who are here, perhaps would love just on a quick overview on really what we're doing here is that we are comparing the clinical and cost outcomes of 2 or more care options. And typically, and as I put it, an easy formula there is really the difference between the net cost of the treatments over the effectiveness of the 2 treatments that we're comparing to. Now, typically we will do these comparisons through a simulated computer model of patients and cost outcomes of different care pathways. And we quantify the measures of gain in health, could be through functional improvement, how this drug may prevent negative outcomes, additional life year gains, improvement in quality of lives. But when all of this are summed up, really to quality adjusted life years and equal value of life year gained. So for this review, our objective again was to, we developed 2 different models for eculizumab and efgartigimod, reflecting the clinical trial that we got the data from. And we used the Markov model with time varying proportions of achieving 3 or more reduction in QMG, health state-based probability of MG-related hospitalization or emergency room visits and mortality. We modeled this from a US healthcare perspective. We used a 2-year time horizon. Again, given the fact that we know that this condition is not a progressive disease and that these drugs work, their effectiveness appears quickly. We used a cycle length of 4 weeks, and again, our outcomes were cost per quality adjusted life year gained and cost per equal value of life year gained. So just to briefly show a few of the model inputs, the health states utilities were derived from a de-identified data set consisting of about 250 patients that was provided to us actually by Dr. Bennett, who is, excuse me, a professor at the University of at Toronto. She does patient-reported research on patient-reported outcome on myasthenia gravis, and we assumed a linear association between the QMG and the utility score we used in the model. Other model inputs, the cost for eculizumab was that was based on the federal supply schedule pricing, annual cost of about $650,000. And we used a placeholder price for efgartigimod of about $400,000 because at the time of this review, efgartigimod was not yet approved. And we made that assumption based on some publicly available information at that time. Based on the costs for MG-related hospitalization and emergency room visits and labor costs were obtained from systematic reviews. So turning to the results, here at this table, if I can just, okay, perfect. So here, this first column is showing the total cost. Again, we conducted 2 different models. This total cost really is a reflection of both the drug cost and some of the other administrative costs over the 2-year period for eculizumab plus conventional therapy versus conventional therapy alone. And as we can see, there's a significant quality gain really with using the eculizumab, but the incremental results show that the cost per quality gain with eculizumab is about $5.2 million per quality gain or per EVOYG. And this really is far higher than the typical threshold of $150,000 per quality gain. We also have the same thing for efgartigimod. For efgartigimod, we have about, again, significant improvement in terms of quality gain when using efgartigimod. And the incremental result shows about $2 million per quality gain or cost per EVOYG. Again, far higher than the typically cited threshold of cost effectiveness of $150,000 per quality gain. So we generally report, and I know Dr. Smith put this up as part of his presentation earlier, what we call a health benefit price benchmark. And I think oftentimes this is the easier part to understand in our results. Really, the question is, based on all what we've done, how much discount from the current price, or what exactly should this drug be priced as? And so based on our analysis, we're saying that eculizumab would require a discount of about 97 to 98% for it to fall within that threshold of being cost effective. For efgartigimod, since we did not have a price as of the time of this evaluation, we just have what the health benefit price benchmark should be. I know, again, now we have a price, and it's, as we can see, it falls higher than what we have there of an annual price of about $18,000 to $30,000. So really the bottom lines of the clinical evidence and cost effectiveness is eculizumab and efgartigimod significantly improve function and quality of life for AChR antibody positive patients. There remains uncertainty regarding the long-term use of both treatments. And especially for efgartigimod, the relationship of the dose and frequency to clinical effect. At the current price for both eculizumab and efgartigimod, they are well above typical thresholds, although the cost effectiveness of efgartigimod appears to be superior to that of eculizumab. So some of the benefits beyond health and special priorities that we cited in the report, just to highlight 2 of them, is that it is important to know that MG affects women in early working lives, leading to reduced working hours, slow career progression, and early retirement. And this is particularly true among Black American women. We also highlighted the fact that MG is a serious lifelong illness with about 60 to 80% of patients not achieving their treatment goals with current therapies. So a few key policy recommendations, just want to highlight, that came from our public meeting after providing this evidence and talking through some of our findings. Was now we have this, we do have a long list in our final report of our policy recommendations. I'm just going to quickly highlight 2 or 3 of this. And really is that all stakeholders have a responsibility and an important role to play in ensuring that effective new treatment options for patients in general and patients with myasthenia gravis are introduced in a way that would help to reduce health inequities. And we mentioned specific stakeholders here. Manufacturers will be important to set the price for new drugs in a way that align with the benefit they actually provide for patients. And it's important to include diverse patient population in clinical trials. For payers, we noted that it would be important to create requirements for out-of-pocket spending that would not create major barriers for patients, particularly vulnerable patients. And for clinical specialty societies, particularly because of what we heard when we spoke with patients and the long path to diagnosis, that it would be important to develop and disseminate educational materials and measurable goals to demonstrate that clinicians are aware of the challenges of diagnosing myasthenia gravis, with particular attention given to providers that care for vulnerable patients. And also want to highlight again, one more, particularly related to the step therapy there, that this was a recommendation for payers that because the cost effectiveness of efgartigimod appears to be superior to that of eculizumab, it might be tempting for payers to want to require step therapy through efgartigimod. But we said that given the limited current evidence on efgartigimod, payers should not require a step therapy through efgartigimod. However, as additional clinical evidence accumulates, it may be reasonable to require step therapy based on price. And finally, I will end on this note. It's again, from the start of our review to we work with patients, completing the evidence review, the public meeting, and some of the policy recommendations that come from there. Now, as part of our process, one of the things we've started doing is to do a 2-year look back at our reviews, and in partnership with patient groups to go back and look at what the coverage criteria, what's been like, and if there's been barriers to access, because we think it's also part of what we do for us to be able to highlight that, because in the middle of all of this pricing we're seeing are patients, are real patients that need to be treated. Thank you again for listening to me. Happy to take questions.
Thank you very much. And we'll invite our participants here to the podium. Mark.
Yes. Good morning. I have a question for the first speaker. Congratulations with the trial. I participated in the earlier trial published by Don Sanders in 2008 in neurology on comparing MMF plus prednisone versus prednisone alone. And that trial, as you know, failed. And now we know why it failed, because we only looked up to 8 months after starting the medication. So that was nice. About the GI effects, my experience is if you dose the CellCept 4 times a day, MMF, instead of 2 tablets a day, you hardly see any side effects. Is that also your experiment? How was the dosing done?
Yeah, that's interesting. I've actually never dosed mycophenolate mofetil 4 times a day. I usually dose it twice a day. I see some GI side effects, but I don't see a whole lot. But that is an interesting point. I'll certainly try it. My question is, in terms of compliance, do patients manage to take it 4 times a day?
Yes, because they usually, if they take mestinon, I advise them to take them together. So that's one point. The second point, of course, in real life, we would like to add another immunosuppressive drug. So this was just a single, of course, in order for the sake of clarity of which drugs is superior. Apparently, there's no real difference. So I plead for the use of MMF instead of azathioprine, because apparently it's more, in my hands, it's more safe. About the second immunosuppressive drug, I mean, I would also plead for a steroid-free treatment of myasthenics. I mean, we have other drugs now, mainly cyclosporin A and even tacrolimus, where you don't need to give the side effect, because if you look at the burden of side effects for MG patients, they mainly related to steroids. And I'm still really impressed how in some countries they go up to 120 milligrams of prednisone alternate day. I think this should be banned from the world and also from our patients.
Thank you.
Thank you.
Steve Riddell from Sydney. I wondered more from the health technology assessment and economics presenters, whether there's been more thought on pushing hard on undoing the Medicare restrictions on drug price negotiations, because it seems to me from an outside perspective that if you have somebody who's selling something and you agree not to negotiate on the price of that something, which is what the US rule does, then that seems to be, to me, the most primary thing that's, undoing your price sensibility. I speak as somebody who's got involved in the HTA process in Australia, but not for drug pricing, so I can comment on this.
Yeah.
Thank you for your question. I want to rephrase that question. Is your question that is there a way in which we could work more with with Medicare?
Yeah, well, as a significant player, if you have somebody who's prepared to negotiate on price, then people will meet you on a price. And Australia has some of the lowest first world drug pricing because our national payer says no sometimes until there's a negotiated price.
Got it. I think the first thing I would say is, again, as you would note, we are independent. We do not have the federal mandate. And so really, we would love to be able to have more mandate to do that, but we can't. And that's why we do have our reports to be publicly available. We always say this is a tool for decision makers to use. We're aware that there are different payers group that use it. We know of some government payers. We know their Medicare organized, like for different states, different New York Medicaid, for example, they've cited using our report. But because we do not have the federal mandate, there's limit to what we can do.
But that's an incredibly important question. And what's fascinating is in the United States, giving Medicare the right to negotiate a price, which makes sense, right? Who on earth goes to buy a car and say, hey, I'm not going to negotiate the price? I mean, it's crazy, right? It's wildly popular. It was part of Build Back Better. We just haven't been able to get it across the finish line. And my sense is that, and others can speak to this, is that from a political perspective, it's a little Sisyphean, but the rock around Medicare negotiations is getting a little closer to the top of the hill. And hopefully within the coming years, we'll have at least a limited availability for Medicare to negotiate drug prices, at least for certain drugs, very expensive drugs.
Thank you.
Pushpa, this is a question for you. Thank you. Wonderful talk. You might have mentioned this, and apologies if I missed it. So the time of onset of the minimal QOL 15 was obviously longer in MMF. Does that include the intention to treat analysis of the azathioprine crossover people? And is there a long time between them? Would it have influenced it?
Yes. So the first question, yes, it is the time to onset of the lowest, from the baseline to the lowest QOL 15 score. So the total time to get to that, not to the lowest 15, meaningful, clinically meaningful, so 5-point drop. So yes, it includes the ITT population, and we did sensitivity analyses where we excluded the ITT population. And so if that changed, and nothing really changed. What I think we have to go back and do, we've got to look at two things, and we are doing them as we speak. So I have to find the median time, not the mean time, and the IQR. And then I have to actually look at the time from when they got to their maximum dose, to their, sort of their maintenance dose, from that time point onwards, and sort of exclude the escalation period and see if there's a difference.
Sure. One small point is that in the gastroenterology world, the azathioprine is often dosed based on 6-thioguanine metabolites. I don't think it's commonly done in neurological practice. And have you looked at anyone doing that, or looking at even surrogate markers like lymphopenia, macrocytosis, et cetera, and see whether they have adjusted the dose based on that?
So are you talking about TPMT screening?
No, TPMT is prior to starting. So I'm talking about 6TG screening, which is to look at the dosing afterwards.
During. No, I don't believe, I don't do it, and I don't believe any of the investigators in the trial have done it.
Mike?
Hi, Mike Hare from Vermont. First question is for Dr. Agbola. So can you teach us a little bit about the ICER process of when you'll come back to myasthenia as more drugs are sort of right on the precipice of being approved and the pricing has now been released for efgartigimod? Is that at the 2-year review, or do you come back sooner if new things happen?
So we do have a couple of process, like different updates. And if there is a new drug entrance in the market, there is a possibility, again, depending on the timing of when this drug is coming down the pipeline. Like I said, we do want to time our review right around the time of FDA approval so that our report is available for decision makers. So if there's going to be a new drug entrance, there's a possibility to do that. We also do have a 1-year evidence update that we do, which of course will be 1 year from the time we did this review. In that, the way we do that, it's a little bit different in terms of we reach out to various stakeholders that we've worked with, including manufacturers, clinical experts, if they're aware of any new evidence that could inform what we have in the report that's been updated that we should update in the report. And then the 2-year look back, I said, is really to give time for this market to end, those drugs, especially if efgartigimod is new, to enter the market. And we can then do a 2-year look back. This is a different project. It's really more coverage criteria and to see how the access for patients looks like. And to highlight potentially if there are things that we consider are not fair access, perhaps if there are coverage criteria that does not align with the clinical trial eligibility, for example.
That's great. We use that with all of our residents now. Your report is mandatory reading. Dr. Smith, the second question is for you. So I was wondering if you might consider changing your definition of cost in your value equation to include burden of therapy and side effects.
Yeah, I mean, I think that, yeah, it's a fair point. That's part of value, right? I mean, that's an outcome. So yeah, I mean, you could put that under non-financial cost or lower quality of outcome. I agree.
But it's never calculated in any of these algorithms. If I use steroids, God forbid, and give them diabetes, they're now treated for the rest of their life with diabetes. If they fracture a hip with osteoporosis. And none of these factors are being modeled in any of these systems. And that's, to me, a real concern.
Yeah, maybe you can you speak to that? How the side effects of medications are factored into the comparative analysis?
So again, part of our goal is to be able to factor that in. There are situations in which we can do that well. There are situations in which it's not well captured because it's not well captured in clinical trial. But typically, again, what we try to do is really to think about, even from the clinical effectiveness side, what the net health, and thinking about both the benefit and the side effects, and hopefully be able to translate that into the model. But oftentimes the limitations, especially for side effects that may be long-term, to that is that we don't have long-term safety data as at the time of this review.
And that's the problem with the US healthcare system, is that we don't have a central database, unlike many parts of Europe, Canada, for instance, where if we use those data, we may be able to obtain the total impact of our therapy on the overall health of the individual.
Yep.
Jan.
Thank you very much for this very interesting presentations. I have a question for Pushpa. I understand first that it takes about a year, 1.5 years before the maximal effect was reached for both drugs. And also that the adequate and non-adequate dose, in fact, had almost the same effects. So is there a possibility that we are looking at the natural history of the disease and not so much of the effect of the drugs? How much do they contribute really to the improvement over such a long time?
Yeah, I think so. When you look at it, there's a couple of points, right? About 2/3 to 3/4 of patients receiving either drug did well, really well. So, and I think that is fairly reminiscent of other data that we see in registries. Don Sanders' registry from Duke, for instance, that he presented data, 73% of his patients, achieved. That was a different outcome measure. It was minimal manifestations. So I think it's sort of reminiscent of what happens, and that's important as a take-home point to realize that a large proportion of our patients actually do well on these therapies, including side effects, because we captured the CTCAE as well, and not a whole lot. I showed you the numbers that had the most severe side effects. So that's one thing. The second is, in terms of what the dosing. So I don't know whether our definition of adequate and inadequate is right or not. I think these patients got better. I don't think that this is a natural history because again, it reflects other data sets as well. I think they got better. But and also the median duration of illness was short. I mean, these people came in early into their illness. They were drug naive patients. And so we know that experience as well. So that reflects other people's experience, that patients who do well are patients who do well within the first 2 years. So this is a population. I don't think many of these patients were refractory. I don't know what happened to the other 25% or so who didn't do as well within the 3 years time frame. They didn't achieve the measures. But these are patients who you hit them hard and you hit them during that first 2 years, they do well. So I think that's the population we've captured. I don't think we are looking at natural history. I think what we are looking at, it's sort of confirming in a way, if you will, in a prospective manner, this sort of intuitive or data knowledge that we have from retrospective cohort studies and other things in the past.
Okay, thank you.
Shri?
Great presentations, everybody. As a clinician, I was thinking about how do I translate some of the lessons from this session in my practice. One of the questions that I don't know how to address it personally, is that we don't measure in ICER review and other things. We don't actually measure time to improve as a metric. And so, for example, for the 2 studies that ICER used, there is a time to improve because of the randomized control data. We know how long it takes for them to have a peak effect. Contrast with Dr. Narayanaswamy study, it looks like it takes a long time with the traditional drugs, perhaps not expensive, it takes a long time for the patient to improve. And I wonder if ICER or other organizations would actually do a sort of a comparative evaluation or analysis of this time to improve so that we can actually share that data with our patients. Say, let's just say off the top of my head, 1 point improvement in MGQOL per month is expected with this therapy, 0.5 point improvement in MGQOL per month is expected with this therapy. That would actually allow us to put it back to the patient and say, this is how these things might work. Sorry, I don't have a question.
So,
well, anybody can answer it.
So I will actually try and make a comment on that. All of the modeling that goes into these cost effectiveness is based on some comparative data, and we use data for IVIG, as you said. We used Dr. Barnett-Tapia's paper. The problem is that your eventual cost effectiveness model, that you have, that the result that you get is quite highly dependent on the data that you use for comparison, number one, and the models and the assumption you make based on the data. So, and if you go and actually look at the ICER review, all of that is described very much in detail. And so I think this is really just the beginning of the conversation, just to bring this into focus. What we really need is more data in terms of comparative interventions that we can then apply to newer drugs that come and look at these parameters.
And I think, thank you, very good answer. We rely on the data out there, and so we throw it back to the researchers to do it. But the other thing I would say about the result for the eculizumab and efgartigimod is they appear to actually work immediately. And so it's unlike some of the other drugs that you talked about, that it seems like there's actually time that you have to give to. For this model, we did not consider, because you're right, if we were thinking about the conventional therapy harm of it, like how long it actually takes.
Because, yeah, I mean,
exactly.
For clinicians who see patients, one of the key factors that we have to talk to the patient is how long will it take for it to work? Without that data point, it's very difficult to translate the cost to the.
But the other thing I would add to it, particularly for this model, and I think for many of these drugs, they're used in patients that have been resistant to treatment. So in a sense, it's that time hopefully has already been given on the conventional therapy. And now it's the question of, should I now be having this new treatment or not?
Yeah.
So did the analysis factor this in to the extent that you looked at the potential reduction in cost of hospitalizations, right? That would be approximate negative impact prevented. So that's indirectly getting at the point that Dr. Mapidi is speaking about, isn't it?
That is correct.
We're a little bit over time, so let's take a couple more, sweet.
Thank you so much. I've got a question and a comment to put that question into context for Dr. Narayana Swami. Love the work. That's brilliant. It was very interesting about the high proportion of ocular myasthenic. The question is, were you able to look into the proportion who converted to generalized myasthenia? The context is patients oftentimes, when we make the first diagnosis, we have to counsel them about seeking medical attention should it generalize. And it invariably causes anxiety, and having a way to explain about the tempo, and also my feeling is we can stratify the risks of those at higher and low risk. And also really within this MGFA community, there are great registries. It's an opportunity for us to work together to pull the data, do external validation, and look into this further. Thank you.
Yeah, absolutely. Thank you for your comment. Yes, the answer is yes. We don't have the answers yet because we are still doing these exploratory analysis. So one of the things, obviously, we want to look at is what proportion generalized. We also want to see how many were treated with steroids and whether that in some way affected the generalization. We may not have enough precision, obviously, but I think this is still data that will be useful. Yeah, we're looking at it. Maybe next time.
One more, Carolina.
So I'm Carolina Varnett from Toronto. Thank you for the panel. When part of the committee, we really wanted to have the discussions. I think we're all worried about this, and going forward is something that is just going to keep coming. So one is a call to arms for the community. We cannot have reliable models if we don't have the data. And this is when we came with the health utilities, because there were none. But there's a lot of data that we do not have. So this utility, what happens with side effects, I know Mike Aher is working into that, cost, the burden of patients. There's a lot of data that we do not have. But also modeling, you can do modeling to look at uncertainties, right? So you can model and look which areas we need more research. So we just need to move forward, because this problem will just continue to grow. This is not going to get away, and we can only help make informed decisions and improve care if we have the data. So I just wanted to thank you too for such a great talks, and to contributing for this too for all of us. Thank you.
Thank you,
Thank
Carolina.
you.
Since I've been calling out on you since all morning, I've been talking about your data and your work.
So I'd like to thank all of you on the panel for your comments and discussion. Want to thank all of you for your questions and your attentiveness to this. And Carolina stole all my comments that I was going to make at the end. So with that, thanks again, and let's go have lunch.